Okay, good morning again. This is lecture 7. Welcome again. So this lecture will be focusing on the control. The last 2 lectures we talked about DNA replication and the process, replication, transcription, translation. And the last lecture we talked about the regulation and how you can accurately control when to produce a protein, and so that to support the function of the genes or support the growth of cells. And so in this lecture, we want to focus on, the accuracy, right? So the last 2 lectures, we assume everything is very, very accurately produced or following the instruction. Okay, and while one analogy you can think about is, you actually none of us actually do it, and we watch them on TV. So imagine you get a blueprint and start building a house, or maybe you've had the opportunity to assemble a bookshelf that comes in a box in pieces of wood that you have to assemble. All right, we bought a bookshelf, what they call modular bookshelf, or it's very has these different shapes, sizes in the bookshelf for display. And so it took us a whole day, my wife and I, a whole day to assemble it together. And the key there is you have to do it in the correct sequence. And so building a house is even more complicated, right? And so, for example, we use a light switch. We talked about lights, turn off the lights, turn on the lights. I'm sure you've all walked into a room with lots of light switch. We were in person, we'll be in Science Hall 1405, which is a fancy room, has lots of switches. Okay, now most of the switch has no label on them. Okay, and so you walked in, and then you need to turn on the lights, and or turn on and turn off specific lights. Then what do? Anyone? You walked into a room with lots of switches, light switch. Then you want to turn on the light of, yes, go ahead.
It would just turn off the one that it's for. It wouldn't turn off all of them.
Right. So you would want to so your strategy is either you turn it on or at the same time turn on all of them and then start turning off each one of them, right? To see which switch is turning off which light. And so then you can end up controlling, okay, so now I make a mental note and say, okay, this switch, number 1, is turn off the light in front of the podium, because that you don't want the light shining on the whiteboard or blackboard. And so in the molecular genetics terms or biology terms, by turning off a switch, turn a switch to turn off a light, that would be similar to, anyone? A term that's often used about genes, say knockout or mutagenesis. So when you hear about the word knockout mouse, that's often associated, is that they have a normal wild type mouse, and they just destroyed one of the genes of hundreds of thousands of genes in a mouse genome. And then they look for changes or behavioral changes in any process right? And so then they know, okay, this gene is controlling this, right? So in bacteria term, it's called mutagenesis. And so you actually turning off, you mutagenize a gene, and you look for a loss of function, right? And so that's in general term. That's assuming everything is doing all the translation, transcription, DNA replication is all done accurately, and that you want to find how many what genes is doing what. And at this point, I also want to mention your best friend E. coli, the genome, the size of genome is 4.6 million base pairs. How many genes typically, we said last time we said a typical bacteria gene is about 1,000 base pair long. So based on that estimate, you will say, well, E. coli can potentially encodes at least 4,000 genes. 4,000 genes, right? And so to know the function of each one of them, and how would you, and so you start from the beginning, you start mutagenize it, and then find the function. Okay, and so that's the problem that we're trying to solve. And but also we're trying to see what happens if there's a problem in the mutation, in the translation, transcription, or in the DNA replication. That's where the mutation will have an effect. Okay, so that's a quick overview of the problem we're trying to solve. So let's go to the detail. So we'll be talking about mutations, recombinations, and bacterial genetic techniques. And then we can talk about how we can control bacterial growth. So here are some of the concepts. I want to go over them quickly. And there's a mutation, and then there's a mutant. In these two terms, it needs to be very, very clearly used correctly. And so mutation is a change in the genetic material. Notice I said it's a genetic material because some organisms use RNA instead of DNA. Right? So mutation is a change in the genetic material. A mutant is an organism that carries the mutation. And the molecular basis for mutation, we typically refer to change in genetic material, and which will come to in a second. So let me first go to the mutation and and the mutant. So related to mutation, then because when you have a mutation, you have a change in the gene. And so how do you tell other people, or how do you mark this there's a change? Okay, so here are some standard way of marking them. Okay, so the first is a genotype. You label bacteria based on its gene. And in this case, it's HIS, is related to histidine. So typically, it's a histidine biosynthesis gene. His C is a specific gene. So between the difference between his and his C, his refer to all the genes that's involved in making histidine, because there are more than one genes, right? In this case, it could probably have his A, B, C, D, E, F, G, right? Depends on which one. So his C is a specific gene. And when you see a number after the gene, it refers to a mutation. So his C2 is a mutation. And his C2 is the second mutation that's isolated. Okay, so each time when we isolate a mutation or identify a mutation, we'll give it a number. And then we actually get it down to figure out what the actual changes is. And then, so that's the genotype. And then the phenotype is easier. That his C plus means it's capable of making histidine. His C minus is a bacteria incapable of making histidine. Okay, and so let's take a look at the molecular basis for the mutation. We mentioned, or I mentioned earlier, it's changes in genetic material. Okay, and so the first thing is in an example of how do you find mutation and a mutant. Okay, so later on you will do the experiment to put antibiotic disc onto a plate streaked with bacteria to measure the antibiotic resistance or sensitivity of a bacteria to a particular concentration of antibiotic. So you will put a paper disc. You will put a paper disc here in the middle, and the paper disc contains dried antibiotic powder. And so now the powder antibiotic will dissolve, and then antibiotic will diffuse out, right? And so the concentration of antibiotic will be high here and low here, right? And so you will notice that majority of the bacteria will grow to about here and then stop. But there will be a few. In this case, you have this, and then you have one here. So these are the mutants. These are mutants. These are antibiotic resistance mutants. And there is slightly different of, so this bacteria is more resistant to antibiotic than this bacteria. Why? Because this is closer to the disc, because it's a higher concentration here than this. Okay, so that's a mutant, and we know they have a mutation, and later on we'll figure out where the mutation most likely is based on the antibiotics. And then if we were to look at the genetic changes, possible genetic changes. So first we can look at the wild type, natural, and you have normal protein, and it's and in this particular part is we're looking at this one particular DNA sequence. Okay, so it's TAC. It's TAC. So TAC gets replicated, normal replication, replicated and transcribed into UAC. And UAC is a codon for tyrosine, so that's okay. However, you can have a mutation during the DNA replication. A C gets changed, a G when you replicate, right? And G gets changed. I'm sorry, it's T gets changed into. I'm sorry, it's C. CG gets changed into TA. And then it gets transcribed into UAU, and luckily it's still tyrosine because the third codon wobbling, remember, the third codon wobbles because there are 4 codons. So the tRNA is tRNA, the anticodon and the codon matching is more focused on the first 2. The third can be more flexible. And so in this case, it still calls the tyrosine, and so you have a silent mutation. Okay, it's making the normal protein even though the DNA has changed. So it's a silent mutation because you wouldn't detect it. However, if this TAC gets changed into TAG and transcribed into UAG, and last time when we looked at the codon table, you remember you have to memorize the start codon and the stop codon, right? And so it's a stop codon. What does it mean, stop codon? Well, you translate it to half to the middle of the gene, and you run into a stop codon, right? So translation stopped, transcription completed, but translation stopped. Translation stopped, you only have half of the first half of the protein, so it's an incomplete protein. And you have, in this case, from bacteria, the bacteria doesn't have a functional protein. So it's a nonsense mutation. That's the definition of nonsense mutation. Okay, and then you have, you can also have a mutation that changes from TAC to AAC. Now it changes into asparagine. And so now the protein that you have is different, have one amino acid different from the wild type protein. And so this protein is not going to be functional 100%. It's a faulty, right? So something damaged. It's a damaged protein. It's faulty protein. So this, then we call this as a missense mutation. Okay. So that's just changes on base pair alone. But and that changes usually takes place in these mutations usually takes place in the replication. Okay, but what if there's other mutations? You can have a normal. This is a normal frame. Right? This is a normal DNA in the middle. So this is normal DNA and gets transcribed. But if you have an insertion mutation, and if you just insert one base pair, so in this case, it's inserting one base pair, all right, and you will see from CCU to CCC. And based on codon wobbling, maybe this still is, this mutation, this site is still okay. However, if you look at this codon, goes from GUU to UGU, it's completely certain that this amino acid will be changed. So an insertion mutation changes, and this changes the frame, right? So it goes, because there's one extra C here, and the U gets to here, okay? Extra C here, and now the U moved to the next frame. So the net result is every protein or every amino acid from this point down is going to be wrong amino acid. Okay, so this then this protein, this bacteria will have a frameshift mutation, will not have a functional, completely completely non-functional protein. You can also have a deletion of one base pair, and that have the similar devastating consequence of changing. You see, it's from CCU to CUG. And so from this point down forward, every amino acid is wrong. Okay, so the entire protein is wrong, is completely wrong, and you would not have a functional protein. All right, and so that's the molecular basis. And so we talked about base pair changes and frame shift and deletion. And what's about spontaneous mutation? Well, spontaneous mutation is something that takes place spontaneously. Okay, so let me step back to think about. So you guys type really fast, right? And you guys, I think your generation or young people can use 2 fingers to type on the keyboard. These tiny keyboards and going really fast. But do you make mistakes when you type? Texting. I assume you do, right? Now, sometimes, yes. Okay, cool. Now, imagine if someone is next to you, even you're the best typing skill. If your best friend is saying, okay, come on, let's go, let's go, let's go. But you say, no, I have to send this to my best friend. Say, no, I'm your best friend. Let's go to what, movies or I don't know. But the point is, if someone is pressure you, and then your typing skill will, the accuracy will increase or decrease. No? Yeah, your accuracy will decrease. That you will make mistake. So the frequency of mistake will increase dramatically, right? So spontaneous mutation is when bacteria is happily growing in the best condition. So meaning that you sitting under the sun sipping coffee, whatever the best drink your favorite drink, and relaxed, and then you typing. And say if you type, your frequency of mistake is 1 in 1,000, right? And then you compare that to your friends pressuring you, then your mutation, then the error rate is, let's say, 1 in 10, right? So now by comparing this, your frequency mutation rate actually will increase, right? So this, in similar analogy, you look at this and say, okay, the bacteria is happily growing, and the highest accuracy is the mistaking, how often they make mistake is 1 in a billion. Can you achieve that frequency, 1 in a billion? You're typing 1 billion characters, and you can only make 1 mistake. And that's just how accurate, right? Now, and then since a gene is 1,000 base pair per gene is 1,000 base pair. So each gene is 1,000 base pairs. So now the frequency per gene is increased, right? Because it's 1,000 genes that you the per gene is increased, and so I'm sorry. All right, so let me step back. So we're talking about the frequency, and the spontaneous rate per base pair is 1 in a billion. But because each gene is 1,000 base pairs, and the frequency per gene is 1 in a 1,000-fold increase, right? And so your frequency per gene is 1 in a million. I know sometimes it's difficult to comprehend this concept. Okay, so the next week, the lottery, mass milling or something, mass milling gone on to 100 million. So now you really want to get that lottery. How do you increase your chance 1,000-fold? You buy 1,000 tickets, right? That's because individual ticket has the fixed rate. But if you buy 1,000 of them, your frequency just increase 1,000-fold, right? Okay, so that's how you that's the difference, all right? So in a net result is that if you have 100,000 gene, 100,000 cell, 100 of them will have a mutation in a gene. Okay, and maybe have more. All right. So how many you've in the lab, right? The professors provide you with liquid culture, right? And if it's cloudy, E. coli culture cloudy, the concentration of that cloudy cell culture is about 100 is 10 to the 8th. That's 100 million cells per ml. Are you impressed? That's just the number. Okay. Now I'm gonna pause just for a second, for a minute. The frequency often gets throw people off. Are there any questions? Is it all clear? And this will show up on the exam, because this is a very fundamentally important concept. Anyone? Question? Okay, I either lost every single one of you or I'm super clear, but I assume I lost everyone. Let me just go quickly summarize. Okay, so the mutation, we said the last week, the replication and the DNA proofreading function of the DNA polymerase 3 ensures the accuracy, and because these accuracy, the spontaneous mutation rate is 1 in a billion. And but when we measure it by per gene, because each gene has 1,000 base pairs, and the mutation rate per gene is 1 in a million, right? 1,000-fold higher. And based on that calculation, if you have 100 million cells, each one of them, if you have 100 million cells, you will have 100 of them have a mutation. And this is an important function that we will talk about. Okay, so now we're thinking about spontaneous mutation and mutagenesis. Now, earlier I said your friend getting you to run is causing you to type wrong, right? Texting wrong, send out the wrong message. And so that's in the biological term, it's called mutagenesis. So now you have a new way of telling your friends, hey, you are what you're doing is a mutagenesis, and you are a mutagen. I'm just joking, but mutagen. Mutagen is a chemical that increases the mutation. And what are the chemicals? Well, in the biological term. And so it's the analog. So let's take a look. These are the analog. Okay, so you have thymine, right? And that's the normal base, okay? But then you have 5-bromouracil. Don't they look the same to you? Imagine how challenging it is for the DNA polymerase III that had to feel the difference in the dark, and then to be quick too. And adenine is this, and this 2-aminopurine. And so with these analogs, that will increase the frequency of DNA polymerase make mistake. Okay. Now later on, when we talk about the treatment, and when we use AZT to treat AIDS or the HIV, that's analog. Okay, to increasing the mutation rate. So now you know the DNA polymerase III and the analog, and these analog is really trying to confuse the DNA polymerase III so that when they form base pairs, it looks similar. Okay, well, not looks, it feels the same, and it won't feel, so that increases the chance of introducing a mutation. So that's one, and another is the UV, the wavelength. So we know you're going to the beach or you're going to a long hike, you want to put on sunscreens, and that protects you, protects the skin for skin cancer. But what is it? All right. So what does it do if you have a mutation? So the high energy, the ionizing energy can actually, if you have really strong radiation, that actually kills you, right? If you go, remember the Chernobyl nuclear accident, and that was really a nuclear power plant, and that meltdown, and that really killed people, and people trying to shut it down, and the first set of people who went in trying to contain, and they all died because they got too much radiation. The DNA was broken, right? But at the lower rate is so here's the visible light, and the ultraviolet light is here, UV. That's where the UV is, okay? And the UV typically cause TT dimer. TT, when you have two T's next to each other, it gets transcribed. The two T's linked together looks like as one T. And so this is on DNA. And then when it's transcribed, and it just transcribe as one, okay? And so the TT, and so if you go from two T's to one T, or two U's to one U, what kind of mutation is that? We just learned. This is deleting one base pair, right? And what happens when you delete one base pair in the gene? The translation frame will shift, right? So that's a frame shift mutation. Yes, thank you. That's right. It's a frame shift mutation. Then that means the whole gene gets disrupted. So imagine if you are exposed to the sun too much, and then you start losing functional proteins. Now, if that protein happens to be a cancer checkpoint, well, then you have a cancer cell. Okay, so that's now luckily for humans we have 2 copies of genes, right? And so you it's more likely you get less likely you'll get you will have a mutation that's so devastating. But DNA, well, E. coli always have 1 copy, right? And so then what happened? And so this is what happens. And so this is E. coli has a mechanism to repair DNA damage. All right. Now this looks like a complex process, but it's actually pretty simple if you follow it. Okay. So you have the REC-A protein. It's a recombination protein, which we'll talk about very soon. And REC-A protein, it will be activated by damaged DNA. So now you have, so it's a protein, and now it's an active REC-A. So it have REC-A floating around, not active, and now its DNA damage triggers this to be active, and once it's active, what does it do? Well, it cuts this protein, LAX-A. It just cut it, okay? So it cuts this protein into pieces. So REC-A inactivate LAX-A protein. So this LAX-A protein cut into pieces. Okay, so what? Well, okay, what does LAX-A do? So LAX-A protein normally is a repressor, binds to LAX-A operator. We know operator is actually the term for the repressor binding site. And we know what happens when repressor binds to the operator, right? It shuts down. So normally the LAX-A protein binds here and shuts down here, shuts down these DNA repair genes. So now, if you don't have LAX-A, then these genes are expressed, right? And now if this gene is expressed, the DNA repair mechanism is turned on, okay? So how convenient is this? You see, it goes here to here. So if you think about this, if you just look at the reaction. So DNA damage, let's say UV, DNA damage, DNA repair, right? So, and if you skip the middle part, right? So DNA damage, DNA repair. Now, once the DNA is repaired, what happened? Well, you need to shut it down, right? And you need to turn off this process, right? And so how do you turn off? Well, it's embedded in here. So DNA, when DNA damage, when it's damaged DNA, the REC-A is activated. But if you look at this here, there's a REC-A is actually also controlled by LAX-A. Okay, so in normal case, when you don't have when the DNA damage, when damaged DNA is wrong, then this becomes inactive, and this then gets accumulated, right? Because this is accumulated. And when this is accumulated, this actually shuts down the REC-A as well. All right. So when you have so now let me see if we can have. So what happened is UV, right? And then you have DNA damage. And then you have REC-A active. And LAX-A decrease. And then you have DNA repair. Okay. And what's interesting here is Lex. Oh, do I? And this is the part that sometimes people get confused. Lex-A increase. You see, when this turn on, when the DNA repair mechanism is turn on, but the LAX-A protein itself gets turned on, right? And so if you keep increasing the DNA repair, but the LAX-A protein is also keeping increase, right? And so this then is, as soon as your REC-A, active REC-A is decreased, then the LAX-A will shut it down. Okay. Oops. REC-A decrease.
Professor, when LAX-A is degraded?
Say it again.
I see LAX-A is degraded. When is it and how it happens?
It's degraded by REC-A, by the active REC-A. REC-A plus the damaged DNA become a REC-A active, and then digests LAX-A, and LAX-A gets decreased. Okay, and so then, so now you're going to have okay, so that's the switch. Okay, so if the sequence, I ran out of space, but that's how it's done. Okay. All right, let's see if I can move. Oh, I can move. Oh, sorry. Okay, so that's the DNA repair. So we've talked about the mutagen, the radiation, and how DNA can be repaired by bacteria through the SOS system. And now we want to talk about the term of carcinogenesis. What's that? Does anyone know this term referring to?
No, it's like cancerous.
Yes, cancer causing. So if you want to know if your hairspray or mouthwash, whether it can cause cancer or some really nasty chemicals can cause cancer, what would you do? You want to know if it can cause mutation, cause an increase of mutation, right? Well, because carcinogenesis is just a mutagen, right? So that should increase the mutation. That's a problem that was presented to people a long time ago. And then we have the Ames tests. Who created Ames tests? Who created Ames tests? It's a tricky question. But you know, people often name something important after the person who invented that, right? So by that means Professor Ames invented the Ames tests. Okay, so it is Professor Ames, and so so this is a very simple test. So if you look, so what he did was to put the bacteria a let's see, his minus bacteria, and he's looking for his plus. Now, you can have a his plus goes to his minus as a mutation, right? But the his minus mutation can also revert back to his plus, right? You can have a mutation goes from his minus to his plus, right? Assuming that's one base pair change. Agreed? Okay, so then, so he put the his, so his minus mutant on medium with no okay, so if you put a his minus mutant on a media with no histidine, will the bacteria grow? Remember, histidine is one critical amino acid, right? So if there's no histidine, and the bacteria can't make itself, and you don't provide it, will the bacteria be able to grow? It's like, okay, if I if I don't give you if I don't give well, we don't do that, but if you don't give food to a mouse, will mouse live? No. Okay. And we all know there are some amino acids we can't make, right? And so we will have to get it from food. And so if you don't have that, you won't be able to grow well, stay healthy, right? So his mutant on media with no histidine will not grow. Okay, so if you put, let's say, 100. So if you put 100, no, it will not. Oh, yes, I'm typing. Can people see it? Huh? That's strange. Okay, now yes. Oh, okay. I have to click outside. I'm sorry. I'm learning. Okay. So let me so it's a his minus mutation goes to his plus, assuming it's caused by single mutation. So the his minus on a media with no histidine will not grow, right? But if you put 100,000 his minus mutant there, and you're gonna get 100 mutant, right? Maybe I can move. Am I allowed to move this? Oh, I can move. Okay, so you get 100 mutant, right? Okay, so this is what he did. He put 100,000 bacteria on this plate. You know, he just spread it out evenly, and then and then he put chemical on this plate, and then you look and one chemical one here and chemical two. So these colonies, individual colonies, are his plus mutant in this case, goes from his minus to his plus, right? And so now each colony have these, so these each colony represent a mutant. And if you look at this chemical here, you see a lot of mutant, a lot of colony here. And so the chemical, this chemical here, let's say this is 1, this is 2. So chemical 2 will cause a lot of increase the mutation, because there's a lot more mutant around chemical 2. And so that's how you can actually get the chemical. All right. So I know these are a bit more difficult to follow, and so this is what the Ames test is. So I spend more time on the mutagenesis and carcinogenesis because those are somewhat more difficult to follow. And then what if you want to isolate a mutant? And that's a challenge, right? So you use histidine, but what if now you want a tryptophan minus mutant, right? You want a tryptophan minus mutant for similar analysis, and a tryp minus mutant, that would be a mutation in the tryptophan biosynthesis gene, and that is called the auxotroph. And so then in order to isolate a tryp minus mutant, your challenge is the mutation rate is very low, right? So at the best, you'll get 1 in a million. So if you have 1 million bacteria spontaneously, and if you increase the mutation, you still got to somewhere between, let's say, increased, right? Maybe 10 in a million. Right? And so how do you know which one is the tryp minus? So you have to test them. Okay, so let me first look at the how do you actually test which one is tryp minus, so the auxotroph. So the test of seeing this is that what we call the replica plating. So if you put all bacteria, you grow them 10 colonies on, if you put 10 colonies here, and then you put them on a cloth, and this is on the velveteen, velveteen is a specific cloth that has these fibers sticking like needles standing out. And it's actually very difficult to find the right velveteen. I had to, the last time I went to Fordham, Jerome Avenue, Fordham, there's a fabric store. I guess people don't make their own clothes anymore, so fabric store is very rare to find. And so the velveteen, and so you put the colony on there, and you print the colony. Now the colony is on the cloth, and then you put a clean, sterile petri dish on here. One is the minimal media, one is a complete media. So a complete media will have everything, right? All the amino acids. And the minimal media has no amino acids, no added amino acids, right? So a trip minus will not. So the trip minus will not be able to grow here. So trip minus are these, right? And because this is, so if you look at this, the trip minus cannot grow on here because it needs tryptophan, but it grows on the complete media. So that's how you distinct which one is trip minus, right? So that's how you isolate. However, that's 10. So if you have 10 colonies that you can get to it, right? You can test them on. But what about 1 in a million? Let's go back to this part. The challenge here is 1 in a million. Remember? Okay. So in order to reduce the number and okay, let's see. So naturally, it's 1 mutant, 1 trip minus in, let's see, can I do this? Yeah, 1 million trip plus, right? And so you obviously can't possibly do that. That will take forever to put 1 million bacteria onto the plate. So that's not possible. So what you need to do is to get it to oops. So what's doable is 1 trip minus mutant in 10 trip plus, right? So if you get to that, then you can put it on the replica plating to test that. And so in between is where you're going to have to reduce. So how do you go from there? How do you go from that to that? And that's the enrichment, okay? Enrichment is that's the increase the numbers of the mutant. So we do a few things, and so what you do is so you put this trip 1 trip and 1 million trip plus, right? And then you grow them on minimal medium plus ampicillin. AP. Okay. Now we know ampicillin is an antibiotic, right? And so if you do that, what happened? Which one will grow? On this media, which one will grow? Oops, sorry. It's a minus. And anyone? Will the trip minus grow? The trip minus, we just said, won't grow on minimal media. And will the trip plus grow? Trip plus will grow. Now, when this trip plus grow in the presence of ampicillin, what happens to its cell wall? The cell wall is weakened, and they will then die. Okay, so now on this, what happened? Let's see. Oh, okay. Trip plus explode. Lies. Okay. Trip plus cell lies, and they die. Right. And so now if everything is working perfectly, then what's left is one trip minus bacteria, right? However, life is more complicated. You have lazy cells. You can imagine that there are some lazy trip cells that didn't grow. Okay, so now what you ended up with one trip minus and say 10 lazy trip. No, don't use it. The lazy word is maybe I'll here put in parentheses. Okay, it's a description that that's non-scientific. All right, what I'm trying to make you see is nothing is perfect. There may be some lazy bacteria trip plus cell didn't grow. So now you went from 1 in a million, now you go from 1 to 10. If you got to 1 to 10, what can you do? Oh, I need to get out of here so you can see what I typed. So 1 trip minus and then 10 lazy trip plus. Now you can put them on the petri dish. Now you can screen them. And that's how you isolate. So you need 2 steps. You need to enrich, and then you need to screen. Question? Okay, so in real life, this is what they look like. And so this is a replica plating. And so this is the rich media, right? Every bacteria is growing, but you see these, and these. Obviously, you mark on the position so that you can compare. So this and this is the same. So you see missing here. Okay, so now that's how you actually isolate trip minus mutant. And this is very important because this is how we were able to identify different genes, identifying which gene is doing what. right? Without that, we were not able to map. Now you have all human genomes. You can sequence all you want. If you get a whole genome sequence, it's a continuous long letters. How do you know which one is which gene? So you still have to do functional analysis. Okay, many of the current interpretation of what gene does what is all from studies of E. coli and bacteria. Okay, so what we just talked about, spent a lot of time talking about, is mutagenesis, spontaneous mutation, mutagenesis, and carcinogenesis, and how do you isolate mutant? Because I think those are really challenging questions. The next question I wanted to tackle just slightly is, and I won't be able to explain all, is recombination. So you all heard about recombination, changes of DNA, and so what you actually what's actually happening is a bit more complicated. So you have donor DNA and you have recipient DNA. And so what happened is that the donor DNA, when it gets, this is all happening inside the cell. And then the donor DNA gets nicked, single strand cut, single strand, not double strand, single strand cut. And the Rec-A protein, the one that we just talked about, cuts the Lex-A during the, and the main function of the Rec-A, well, the initial function of the Rec-A was first discovered because it's involved in recombination. So you see, Rec-A, recombination protein A. Okay, that's where the protein Rec-A comes in. So what does it do? It takes the strand, comes in here, puts in here. So now, and puts this strand goes up. So now you have a cross strand. So now one strand goes up, one goes down, and then depends on how do you resolve. You have to separate them, right? So how do you resolve? If you resolved in using this, in cutting here, then you're going to have a patch. So one strand of DNA exchange with another. That's one type of recombination. Another is resolving in the blue, and that resolves in the splices. And this splices is the recombination we talk about more. Okay. And so here, the next example I will come back to talk about is that when you think about recombination, typically, if you have one circular DNA and another circular DNA, and then you have a cross over, and so the two circular DNA molecule cross following the splice model, and you go from here and there. And so one single recombination event between two circular DNA molecule turn that into, from two molecule into one molecule. Okay, so if you follow that and say follow here, goes up there, and right, and so see if you go, come on, I have to change yellow, no, red. So if you start from here, if you follow the strand, right, cross, and so if this crosses over here, so then comes back and it crosses back here, it's linked. So is it one molecule or two molecule? It becomes two. So that's one between a circular and a, so now, but now if you have one circular, one linear, okay, so if you do that, so if you're from, oh, so if you have a cross over that recombination event, and so what will happen is that if you follow that, okay, so if a circular molecule and a linear DNA molecule forms, have one recombination event, then it becomes, the circular DNA molecule becomes linearized molecule, a linear molecule. And we know linear molecule cannot be replicated. However, if you have a circular DNA molecule and a linear DNA molecule, and if you have two recombination event, let's see what happened. Two cross. Okay, so from here you go following, go up and come back, and when you have two recombination event, your circular DNA molecule is still circular DNA molecule. The only thing different is that this, let's say, small b, and you have a big B here. The only difference is that it exchanged the genes, all right, replaced its own gene with the gene on the linear DNA molecule. All right, so that's the recombination event. And okay, so so let's take a look at real life examples. In order to look at the real life example, we'll be talking about different DNA genetic techniques, transformation, transduction, conjugation, transposon, and PCR. So for transformation, and this is the experiment that you all have learned in genetics and in introbio, and it shows up everywhere. This is where they prove DNA is the genetic material, right? Where you boil the DNA, you boil the bacteria, you mix them with a bacteria that doesn't form doesn't produce capsule, and then you mix them together and you inject into the mouse and kills the mouse, and the one that comes out is a bacteria that produces. So now all of a sudden, the living bacteria that wasn't produce capsule, now it's able to produce capsule. And the reason is that the DNA from the capsule producing bacteria that was boiled, and so when you boil this bacteria, and the DNA comes out, and so that's what's happening. So the DNA comes out and gets transformed into the bacteria. You see, it goes in here and lines up with Rec A protein, and then replaced. So now these line up this what's here and replace what's here. You see, the end result is that stretch of gene or chromosome gets replaced. In this case, the host, the cell that's being transformed, has a copy of the gene, a damaged copy that's not producing capsule. So now you're replacing it with a capsule producing gene, and that's what that happens. Okay, so that's the basis of transformation. The key for transformation is the DNA, naked DNA on the outside, gets imported into the cell in single strand and then incorporates into the chromosome. So the two steps: when it gets in, when it's into the cell, and then into the chromosome. The next technology, so that's the recombination. I'm sorry. So let me just highlight. So that's where the recombination takes place. Okay, the recombination. The next is the generalized transduction, and I will save this and discuss this at the end of the next lecture. But just to simply point out to the net result. So the net result is the donor. So this is rely on phage to take a piece of the donor DNA to put it into the recipient. So notice this here. All right. So essentially, the phage carries the donor DNA into the recipient DNA, into the recipient cell, and recombined into the chromosome. I'll explain how this is actually happening at the end of the next lecture. Once you understand how phage grow, life cycle of the phage. The next thing I wanted to, very interesting thing to talk about is the conjugation. So this is happens more frequent. And so this is a pills. Okay, this is a fiber linking two bacteria, and the two bacteria are then get closer and closer. And so what happens is that when you have two bacteria and you have a pilus, and one of the genes, the donor DNA, encodes a pilus gene. Pilus is usually made on this F plasmid. And then this bacteria makes pilus and brings in the other bacteria closer. And then they have a channel. And then they have this donor DNA. Now, it's important that you notice here, it's a single nick. Okay, single-stranded nick. And then the donor DNA gets a copy. And you see it goes in there. All right. And so the conjugation of a plasmid is actually the DNA, the donor donates a copy of the plasmid to the recipient cell. So it's not like you just gave a copy. Why? Because remember, if this is so this part here is a 3' hydroxyl group. And so now if you have a 3' hydroxyl group, what will happen? That could be extended, right? So this is extending, and this is going here. And so now this is completely replicated. And then once it gets in there, it gets replicated too. So now you have two copies. So in the transfer, this nicking part is not a random. It's a specific site called ORT, origin of transfer. It's on the F plasmid. Okay. Now the F plasmid also have a insertion element called IS element that has the exact same sequence. And so here is the recombination shows up again. So if you have an F plasmid that normally does just the conjugation itself, right? It breaks up and it moves to the other donor DNA, from donor to the recipient. However, if this plasmid gets integrated, has one recombination event, one recombination event, and integrating to the chromosome, and the whole chromosome now can be moved from donor to the recipient. Okay, so now the F plasmid is in the chromosome and in the E. coli chromosome. This is actually E. coli chromosome, and then the whole chromosome, E. coli chromosome, in theory, can be conjugated to the recipient, from donor to the recipient. Okay, so this is the conjugation. So you see this, the whole chromosome gets moved. All right. Now, in theory, that the whole chromosome gets moved, but usually that only happens. This doesn't stay on forever, and they stay on. They break up. So each time if this is entirely completed, it will take 100 minutes. Okay, it takes a whole 100 minutes for the entire chromosome to go over. However, it doesn't stay that long. In more reality, it's pieces. Okay, it's whole long pieces of donor DNA. Chromosome gets moved to the recipient cell, and then from here you recombined into this. And so the net result is that if you wait long enough, the whole chromosome can be conjugated in, and so this is what's called the linkage map. The linkage map. This linkage map is the first genetic map of E. coli. This was constructed in the 80s, 1980s. That's 30 years ago. That's way before the whole genome sequence and bacterial sequence. Okay, back then sequencing is a very, very challenging work, and as a postdoc, I spent 3 years sequence 2 kb, 2,000 base pairs. Now you get a bacteria with millions of chromosome, millions of base pair. Machine does it for you. That's all in 1 day. But back then, the linkage map is the best we have, and they were able to map out. So starting from 0.0 minutes. So these are really the units. These units is minutes. Okay, so you can probably know that the linkage map, unit of linkage map, or the distance of linkage map, is minutes, the time, minutes. Right? That's because it takes in theory, takes 100 minutes to transfer the entire chromosome from the donor to the recipient. Okay, and so these are the genes on it, and you can find the lac gene, okay, at 8 minutes. The next important elements I wanted to comment on is the IS element. This is the IS element. The basic IS element is a piece of DNA, has 2 inverted repeats with an enzyme, encodes 1 enzyme called transposase. Now, when the IS element is repeated often, and then what happens is they form a transposon. That's called the transposon. And typically, they will have antibiotic resistant genes in the middle. And one of the the transposon gene is actually interrupted, and only one transposase genes is functional. And transposon can transpose in two different ways. I'm sorry, let me step back. For transposon, there are two types of transposon. One is this is one of them. And so typically you have a donor, and the donor transposon here, and the transposon replicate itself, and then it integrates. So this is the reverse, right? You have one large circular DNA molecule, and it lines up, have one recombination event, and this becomes two circular molecule. So if you look at this, the net result is here. The target, now the target with a transposon. So this is called the replicative transposition. Okay, so the replicative transposition is transposon goes from one to the other here. You can express this in a different way. So you have the target, right? And so this is the target. And for the replicative transposition, we said the transposon, you see the transposon here? Okay, the donor, still donor, and the recipient receives a transposon. But in contrast, you have what's called the non or conserved transposition, conservative transposition, or non-replicative transposition. What happened is that the transposon actually goes from here to the donor and leave the to the recipient, leave the donor with a gap, double-stranded break gap. Now this plasmid can no longer be replicated, and then it's lost. Okay. So related to the transposon, and it's very important. I spent a lot. It's a very important tool. So thinking about transposon, right? Jumps goes from one into another DNA molecule, but where does it go? So think about on the chromosome. So you have normal chromosome, you have gene 1, gene 2, and gene 3. Now you have a transposon jumps in here. A transposon, a large transposon jumps into gene 2. Is the gene 2 remain intact? No, typically it interrupts the gene 2. It typically, because it's long enough, it interrupts the transcription. So you don't have gene 2 at all. So you just lost the gene. And why is that important? We took advantage of this particular part of knocking out genes, and we use transposon to do mutagenesis. Okay, so we make transposon into transpose into a chromosome, and then we isolate mutations. Okay, we screen for mutations, we identify which gene gets interrupted, and that's how we identify the function of the transposon. So that's the transposon mutagenesis technique, and the next thing I wanted to mention is PCR, which I assume most of you already know what PCR is, so I'm going to skip that and asking you to read the details. So these are the basic technologies we talked about. I spent most of the time on the more complicated issues. Now I'm going to go over some of the more easier to follow issues and go over relatively quickly. And so want to talk about how do we control bacteria growth, meaning kill bacteria, essentially. And so you have physical and chemical methods and antibiotic. So physically, you can use autoclave. The key for autoclaves essentially is a pressurized cooker, and we pipe high steam, high pressure steam. So it goes to 121 degree and 15 pounds per square foot, and that's where it will kill. It kills endospores. So 121C and kills it kills endospores. That's the okay. And so you would the autoclave takes longer to you have to raise the temperature, and yeah, I'm sorry, there was a question about the quiz 5. It's listed on the syllabus. You should take a look at the syllabus. It covers lecture 6 and lecture 7. Okay, so that's autoclave, how autoclave looks like. Another way of physically removing bacteria is filtration, and based on the size of the pore, you can use filters to remove. And so in this case, a typical sterile filter will have the pore size of 0.22 micrometer. Okay, so that's what we use to filter out the bacteria, because that's a typical E. coli is 1/4, right? 1/3. In terms of chemicals, and there are different types of chemicals, and there are chemicals that you can separate the chemicals into different types. You can have bacteriostatic, so a chemical that basically stops bacteria from growing, but it doesn't kill bacteria, so the viable count's still there. Viable counts is counting the living bacteria, right? Viability, viable is a living bacteria. And then you can have bactericidal that kills bacteria, and then you can have bacteriolytic that actually physically lyse bacteria cell. So there are 3 different types. To measure the effectiveness of the antibiotic, next we're going to talk about antibiotics. So to measure the effectiveness of antibiotics, you do a serial dilution. So you have cells and you have high concentration, high, and you can have low. Oops. And you can have low concentration of antibiotics, right? And as concentration decrease, somewhere the bacteria, the antibiotic will lose the ability to inhibit. And so that's how you determine the minimum inhibition concentration. Another way is to do the paper disc. You put the paper disc onto it, and you determine the sensitivity by measuring the ring. Okay, and that's the distance you measure, and that you have antibiotic resistant colonies. The the more important thing about antibiotics is how does antibiotic work, right? And we already know one antibiotic, right? That's penicillin or ampicillin, right? This one. And we know it blocks the cross-linking of the cell wall and peptidoglycan, so the peptidoglycan cell wall is weakened, and that is that kills the bacteria because they grow and they lyse. But there are other antibiotics, and these other antibiotics are inhibiting other processes. So the best antibiotic is an antibiotic that kills bacteria but has no effect on human cell. That's the best antibiotic, right? Because you target, so you kill bacteria but not kill us. So you target what's different between bacteria and eukarya, right? In this case, ribosome and the translation is the biggest target because ribosome is different, right? Ribosome, bacterial ribosome is 30 and has 30S and 50S subunit. And the eukaryotes, let me remind you, is 40 and 60. Okay, so they are very different. And so now you actually, so these are the antibiotics that blocks the translation. So I would encourage you to recognize at least one antibiotics that can block translation. Okay, so let's say tetracycline, right? And then you can block RNA polymerase. You can also block DNA gyrase, but those are very, very toxic antibiotics. Why? Because the difference between prokaryote gyrase and eukaryote gyrase is very minimal. Okay, so if you take this and the DNA replication of your cell will also be damaged, also be blocked, and so that's why it's very toxic. Okay. Now, related to this then is the antibiotic resistance. And the antibiotic resistance comes in. These are the factors we have to consider. And how do bacteria gain the antibiotic resistance? All right, that's important. So there are three different possibilities. One is the change of the antibiotic, or to change the target. So you either change the antibiotic or you change the target of the antibiotics, right? So to change antibiotic target, you can disable the antibiotics. This is penicillin. And if you use an enzyme to cut the ring, and that disables it, or you can add a modification group that also disables it, right? The next is you can change the target. What do you mean by changing the target? And changing target is really you can use the analogy of this allosteric molecule. If you change this protein, right, and then you can change the binding site. And you put it here, you change the binding site, and then you prevent the interaction. So this has to do with the specifics. So the tetracycline, what does tetracycline do? Well, it binds to 30S and blocks the initiation formation. So it binds. Remember, tetracycline binds to the 30S. So if you have a mutation in the binding site of the 30S, well, then the tetracycline no longer binds. Okay. Actually, the most common resistance for tetracycline, I take it back, the most common resistance for tetracycline is the pumping out. There's a mechanism, there's a pump for pumping out. It's a streptomycin. Streptomycin is actually binds, and the most common mechanism for streptomycin resistance is the change of the 30S ribosome. Have a mutation in the chromosome. So then you can have these mutations. So the single mutation on chromosome could result in the antibiotic resistance. Okay, but most common is the enzyme, or more difficult or more problematic is the, let me rephrase it. The most common antibiotic resistance mutation, the most common mutation is a change of the chromosome. Why? Well, because to have an enzyme that cut the penicillin or modify cannot be achieved by single mutation. So for single mutation, it's typically a change of the target. Is that clear? I know we ran out of time, so please understand that part. And because that's related to the spread. And the change of the chromosome is very unlikely to be spread. It's less frequent. Its spread could be spread by phage, but most commonly spread antibiotic resistance genes are the genes that modifies the antibiotics. And if the genes on the plasmid, then the plasmid can be conjugated, and that's spread. If the gene resistance genes is on the transposon, it can be transposed to the recipient and gets transposed to the recipient. And that's how the spread is taking place. Okay, and so those are the topics I wanted to discuss for today's lecture. I'm going to stop the recording here.